everybody to Paleo Talks. We are on episode 20, believe it or not. We started with Mastodons with Dr. Chris Widga way back in some month earlier this year. I guess it was maybe March or something. But here we are on episode 20, and we're doing Mastodons again. And we're here with Emil Karpinski, and he's coming to us from McMaster's University up in Canada. Hey, Emil.
Hello. Here we all right?
And you're the superstar this week of Mastodons.
Yeah. I mean, it's again a collaborative project. It's always important. Even my title slide, I think, has all the authors. It's not, these projects are never possible alone, right? These are there's way too much information for any single person to keep in their head. So.
And so yes, with that, we'll have Chris Widga with us today as well as our resident Mastodon expert. Coming to you here, we are from East Tennessee State University, the Center of Excellence in Paleontology. At the Gray Fossil Site, we have a 5-million-year-old mastodon that is bizarre, that Chris and I and others are working on. And Chris, for a long time, has been working on mastodons, and I've always been a great mastodon admirer. I know, Emil, I've heard you talk about mammoths and mastodons, and that mammoths, especially the woolies, are often much more popular, but I've always leaned more toward the mastodon myself.
And we need these advocates because, if you think about the relative PR of these animals, I mean, it's night and day, right? I haven't seen a movie with a mastodon yet, like a nice proper one. When they get a cartoon character like in Ice Age, then they'll probably have made it,
Have you seen the desserts, the mammoth desserts? They're kind of like Little Debbie's.
I have no, they're I've seen a box on them on Facebook. Someone linked it in to me when all these the talk footage went up. There's a board game called Mammoth, although it's German, and I think Mammoth is German for.
Of course, there's a
I
band
might be wrong.
too, called Mastodon. There's a band as well.
Yes, yeah. Chris was Grant wanted to see if you could get them to play for the Tuesday panel.
Well, I grew up in Missouri, just over the hill from where all the Missouri mastodons came from. And my grandpa used to say, up over that hill, that's where they found the dinosaurs. And of course, that's sort of the common misconception.
Yep.
And then ended up going to the Illinois State Museum, where Chris also went, where we just live with mastodons all around us.
You're in the shadow. Yeah.
So I feel like I grew up on mastodons.
Good.
You did. That big guy that's in Emil's data set was just around the corner from you.
I know.
Yeah.
I know. Well, we'll take
we'll see a picture of him later.
A brief minute here and let David tell everybody a little bit more about the logistics of the show. David?
Absolutely. So welcome back, everybody, to Paleo Talks. I see, looking at our comments, that we've got some of our regulars back. I think I even saw a couple names that I didn't recognize, so hopefully we've got some new people tuning in for Mastodons. This is going to follow the standard format. We're going to let our guest go ahead and give their presentation. We've got Blaine and Chris both on hand to chime in with questions and comments. And then about halfway through, 25, 30 minutes in, we're going to wrap up the presentation and open it up for audience questions for our guest. And then, like I said, we've got a bit of a mini Mastodon panel here today. So this is a great day to ask your Mastodon questions. When that time comes, we'll ask you to start putting your questions in the comments next to the Facebook video. And as usual, although this hasn't seemed to have been a problem recently, if you have some sort of trouble commenting on Facebook, head over to the Gray Fossil Site Twitter or Instagram account, send us a message there with your question, and I'll keep an eye on that as well.
All right, thank you, David. Well, Emil, if you can go ahead and sort of move over to your shared screen.
Can you see that all right?
Now we can. And before you get going, just tell us a little bit about your background, where you're from, how you got into studying mastodons and paleogenetics.
So I'm very fortunate in that I went to the university here in Hamilton, Ontario, where the McMaster DNA Center is, and it's one of the few ancient DNA centers in Canada. I think we're definitely the largest and most prolific one when it comes to like impact of research. And it's kind of very by chance. I didn't expect to go into ancient DNA. I didn't expect to have anything to do with paleontology. I'm a molecular biologist by training, so I spent all of my time dealing with very small little microbes under microscopes and on plates. But as I was going into third year, we started the iGEM Club at McMaster, which is a synthetic biology competition. And one of the big avenues or the big dreams also of synthetic biology is to kind of bring back mammoths. And I said, okay, well, that's kind of cool. So I pushed really hard to get into Hendrick's lab when I realized that the Ancient DNA Center was on campus at the end of my undergrad. And from there, I wanted to go do mammoth genomics, which is sort of what we did for our 2018 paper. That data was finally published. But at the same time, as you go in, you start working on a whole bunch of different stuff. So I did mammoths, I did mastodons, I did sloths. And this paper, this is one of the earliest projects I did. It was one of the earliest ones that I was trained on. So it holds a special place in my heart. But it's, yeah. And I mean, when I went in, I didn't, again, my experience was very different. I didn't know what a mastodon was. To me, I looked at them and I said, oh, it's a mammoth that looks slightly different in the reconstructions. That was my experience. And then I very quickly soon learned, I'm like, okay, no, these animals are different and they're kind of cool. And I don't have to keep showing these open flat plains with like one animal on them. I can have a bit more, kind of environmental context that they're in.
Well, great. Well, let's just go ahead and start going through. And you're a student there of Poignard, right?
Yeah, PhD candidate now. I'm probably entering my last year, hopefully entering my last year of my PhD.
One of the things that always seems different to me that jumps out is the number of people that end up being on these papers.
Yes, yeah. And I mean, that's one of the things I think when we get to genetic papers, like I said, I know I sound sometimes like a paleontologist, but I'm very much not a paleontologist at heart. I don't have the training. And I think that's really interesting though when we come into this is that you bring in a lot of people. Like on this paper we have, I think Chris, even you have an anthropology degree, right? We have anthropologists, geneticists, molecular biologists, ecologists. Simon is a molecular evolutionary biologist. We have a lot of people all coming in and bringing their skill set, mostly because there's so much knowledge now that we're trying to synthesize in a lot of these larger projects. And so many skills that you need to really do these projects concretely that it's close to impossible for any one person to do it.
Well, the collaborations that we end up forming with these paleogenetic studies are wonderful because they end up including people from all over the world.
Yeah, and I mean, it acts as a nice springboard later for future collaborations, right? Because you collaborate on a project that's really big, and then all of a sudden, you hear just on the grapevine or on side discussions things that other people are doing, and you're like, oh, that's cool, and let's go work on a different project that springs from this one.
But I do have news for you, Emil. You are a paleontologist. You are studying past life using particular tools that might be different than morphology, but you're a paleontologist now.
Yeah, and I mean, and I think that's true. Some of the bones, I don't feel like a real paleontologist because, again, I don't see much in the way of the real bones, right? They're super small when they come to me, and I spend the first kind of week destroying them.
All right, we'll let you get going here.
Okay. So we will be talking today about a paper, but I wanted to start with a very brief introduction, kind of to the Pleistocene in general, a lot of the different methods, ancient DNA, and where it kind of fits into the greater field. So it's kind of, as I was alluding to, when we think about the Pleistocene, when I thought of the Pleistocene before I started this, it's scenes like this. You're talking huge open tundras. You see Beringia all the time, mammoths, maybe some bison, some horses. This one has less ice than some of the other ones I've seen. Some of them have mammoths coursing across glaciers, in snowstorms, which is probably not the most realistic. But this is what most people imagine when we tell them, we worked on the Pleistocene, or as it's known as the Ice Age, right? They know this, and then they know that a lot of these animals go extinct, that they disappear about 11.7 thousand years ago, right? Most people will argue probably on hunting or climate if they're slightly more informed, but these are kind of the two big points that most people have in mind. But a lot of people, once you start talking to them, they have a rough idea of how we do this, but it's, I think, important to stress really, when we're building these things, a lot of these projects, a lot of their fields don't stand in isolation, but they're highly conjoined and they give complementary evidence that we try to synthesize in each subsequent paper and build up on. So these could be things like glacial limits and environmental reconstructions, the actual, of course, excavation of plant and animal material so we can see what these environments were, what animals coexisted with one another. And in my case, what we'll be talking today is mostly biomolecular approaches and in specific ancient DNA. which is a way of kind of looking directly at particular animals and their biological makeup. And it's a relatively newcomer on the scene. I mean, the first ancient DNA sequences, I think, are less than 40 years old. And the field really only hits its stride in the last 10 to 20 years when we really adapt a lot of these methods to next generation sequencing, or this really high throughput sequencing. But it allows us to ask very interesting questions, ranging from what I would consider kind of more basic questions to more complicated ones. So this first one, for instance, is a paper, one of the first papers I published looking at paleofeces from Bashan Cave with Jim Mead. So you can see here, what we did in this paper is we extracted DNA from this paleofeces to kind of confirm paleontological expectations that this was a mammoth that actually laid these, since I don't think there's been any bones found in Bashan Cave. But what we also managed to do is, because we had the DNA, assign it to probably Mammuthus columbi and to a particular mitochondrial haplogroup. So kind of this broader group of animals that are related via the matriline. You know, this can also be expanded, though, relatively quickly into these really complicated or really nice views of how animals moved and how populations were replaced through time. So this is a paper by L in 2013 looking at mammoths pre and post LGM and kind of tracking them at different time slices. And what you can see here is this kind of movement of mammoth mitochondrial groups from North America all the way through Eurasia prior to their extinction. So it looks like we have this kind of back invasion from North America into Eurasia. But ancient DNA also gives us a lot of resolution when we're trying to resolve these taxonomic issues. So you can see here on this slide, it's a recent paper we published looking at sloth taxonomy and evolution, sorry, where the kind of genetic tree is very different than what we recovered from the morphological tree. And what Fred, the lead author on this paper, argues is that a lot of morphological evolutionary relationships among sloths are complicated by an abundance of these very convergent traits. And if you have really good samples and really good data, and you can recover full, nice, high coverage nuclear genomes, you can ask really kind of deep questions about how these animals are related. So not only even dating splits between when animals might have emerged based on genetic records, but you can also look at levels of gene flow between different species, between different populations, and so on. But if we're talking ancient DNA in North America and on North American megafauna, you might have noticed that, you know, in all but one case on that top paper, and that one being sloths was a bit of an exception, we're really talking about animals like mammoths and bison, which are the kind of two biggest studies, most well-studied species among North American megafauna. Horses probably thereafter now. So kind of from a genetic perspective, you know, we have this picture of the field that looks a little bit like this, where we have, you know, a pretty good understanding of the genetics of a lot of these steppe tundra megafauna, but a pretty blurry view of animals in other environmental contexts. So what we wanted to do in this study that we'll be talking about today is start clearing up, shedding some light on these more wooded wetland megafauna by focusing on one of the mammoth's relatives, the American mastodon, which you can kind of see here nicely clearing up some of this image for us. You can see some more American mastodons hiding out by the edge of the water over there in our cleared up section. Now, the Great Fossil Site is famous for its mammoths, of course, so I'm not going to go too much into, we'll probably talk about more what a mastodon, mastodons. They're famous for their mastodons as opposed to their mammoths. So I won't go too much into what a mastodon is, but if you haven't seen them, they look more like this. They're an earlier relative in the Proboscidean tree than we have in mammoths. And unlike mammoths, you know, which we saw like in the first few slides are these steppe tundra animals, mastodons are much more at home in wooded, wetland environments, when we tend to think of them more as browsers than grazers. And there's been kind of a lot of paleontological studies which have come out throughout the duration of when we were working on the genetics of these animals that suggest they have very different paleontological responses environmental responses than what we would expect in mammoths. So on the left, you can see a reanalysis of the radiocarbon record of American mastodons in Eastern Beringia, which found that all of those finite radiocarbon dates that we had in that region were probably false, probably some sort of contamination. And a lot of those ages are non-finite. All of those ages are non-finite, leading the authors to argue that the mastodon habitation in Eastern Beringia is probably not in the glacial region, but it's probably restricted to interglacial conditions. This next summary, which I've modified from a paper by Chris Widga in 2017, looks at mastodon and mammoth habitation in the American mid-continent as in response, at different time slices in response to deglaciation. And what you can see is that mammoths are super abundant in this region during these more glacial conditions, where we expect that a lot of the surrounding region is also more steppe tundra-like, and mastodons only start moving in when these glaciers start disappearing, the climate starts warming, and probably there's an abundance of trees moving in and so on. So with that in mind, there's always kind of a few goals you lay out at the beginning of the project, and they modify as you accumulate data, as you see what kind of questions you have the power to answer. But one of the main ones that you'll find in every genetic project is to increase the amount of genetic data available, both for your own projects, so you can answer the questions you're interested in, but also as kind of as an extension for future studies, that other studies can kind of use your framework to answer their own questions. The next, because we were building this framework, we wanted to see, what it could tell us about different mastodons, how they're related, both across space and through time. And lastly, we wanted to see how they were, if we had the resolution, how they were responding to things like climate change, glacial cycles, and so on and so forth. I mean, ideally, you want to answer always the questions about terminal extinctions, but those are very difficult questions to answer that we've been arguing for decades. Now, most ancient DNA studies, if you want to begin them, you need to acquire, of course, bones. And if you want to do these really large-scale continent-wide distributions, you need a lot of bones. So this study, we had 122 remains that we collected from, I think, over 17, 18 collaborating institutions. Our samples span almost the full range. So you can see they go all the way from across the continent, from the top of Alaska down into central Mexico, which is pretty close, because I think the southernmost point the mastodons are recovered from is in Belize, somewhere around there. We do have kind of some patches within our data where we don't have great representation. Part of this now, is interesting because we have Mammoth pacificus in this region, which would have been great to kind of include in our study, but it gives us an interesting avenue to pursue in future studies. Kind of before we go further into the project, though, we've talked about ancient DNA, but I think it's important to remember, what exactly ancient DNA is, what we're dealing with in these studies, and kind of contextualize some of the results that we're getting. So this is just a very quick, dirty synopsis of what happens to animals when they die. So as in the case like now, in the case where we're alive, in the case of mammoths when they're alive, a lot of their genetic material exists in these very long fragments. So if you think of us, we have our chromosomes, we have our mitochondrial genome, which is all of this very nice circle, large fragments. And it will exist like this because we have repair mechanisms that will repair any damage or breaks in our cells, as they occur. But when we die, a lot of those repair mechanisms stop. And on top of that, we're also invaded and begin to be broken down by bacteria, both from our environments when we're buried, also from our own bodies, which starts breaking us down and using that as energy for its own growth. And what happens as part of this process is that the DNA that existed in very long strands becomes progressively fragmented and becomes progressively shorter and shorter and shorter. And on top of that, it also undergoes chemical modification. So this results in not just shorter pieces, but pieces also that have way more damage in them. So this could be things like bases missing, bases that have been changed, or DNA that was normally double-stranded, resulting in these kind of single-stranded overhangs like this. And then when this animal is, of course, recovered, we have this next batch of introduction. So we have contamination that's introduced anytime we work with the bone. So this will be both as part of the excavation, as part of the transportation, or even in the lab, even though we're wearing full clean suits. And I can show you a photo later if you're interested of the kind of getup that we have to work with these in. But even despite all of that, there's always going to be some contamination that's introduced into the bone. So what we're left with is a makeup that went from these very long strands of DNA in our source animal to something in the bone that looks like this, where we have a little bit of source DNA, we have a lot of bacterial DNA, and we have some contaminant DNA in there as well. And when we get this bone into the lab, or a piece of this bone into the lab, the first thing we have to do is break down a piece of it, typically a few hundred milligrams, and turn it into a fine powder. And then at this point, it's still, bone, it's still essentially all the DNA is bound up. So what we have to do is break down both the mineral and any remaining organic parts of this bone and release that DNA into solution. We call this a supernatant in the field. You can think of it as sort of a bone juice. It's just a mixture of all the kind of bone components, but now dissolved in kind of an overall slurry. And it contains, the DNA that we're interested in, but also a lot of impurities that we don't want and could restrict our future analyses. So what we do is we bind this to a column and typically wash it a few times such that we end up with relatively pure DNA after this is all done. Now, because we have all these additional sources of DNA in here, so we have our DNA from our animal of interest, which is in black here, but we also have our contaminant DNA, both from human origins and bacterial origins. If we sequence all of this as it is now, which we can do, it's very expensive, and typically only a very small amount of that DNA is usable. So what we try to do is increase the relative proportion of our DNA of interest in. So in this case, that was Mastodon DNA, and DNA were less interesting to us for a lot of these questions. We do this via these enrichment steps, which if they work well, you can see changes the relative proportions of these things such that our sequencing costs goes down, our analysis time goes down, and it really helps with these very degraded samples. And at this point, if everything worked well, we get send it off for sequencing, and you kind of sit there for 2 weeks or so that it takes the data to make its way to the sequencing facility, get sequenced, and come back to you, and kind of crossing your fingers that everything up until this point worked well, because we're really just dealing with clear liquids, that you can actually answer the questions you're interested in at the end. And kind of the first step when you get data is turning these short kind of fragmented reads into these larger genetic constructs. So, you're getting reads that are maybe 25, 30 base pairs. If some of the better preserved material from the permafrost, maybe you're talking 100 base pairs. And you're trying to reconstruct either mitochondrial genomes, which are about 16,500 base pairs, or nuclear genomes, which could be billions of base pairs, right? So you're taking all these small reads and trying to assemble them into reassemble back those long genetic contigs. And you can think of this as sort of a puzzle. What we do is we typically take what's known as a reference, which is a guide that we use to kind of reassemble it. And this can be, if you're lucky, either an animal of the same species, or if not, a very similar species. So in this case, you can see these reads kind of symbolized by these little pieces of proboscidean, chopped up proboscidean here. And our guide in this case is a closely relative, the Asian elephant. And what we do through a variety of computer programs is try to map these back on to regions of our reference that are going to be similar, probably not exact, but similar, and piece together to recover, our complete image of the source organism that we were working with. Now, typically you'll do this a few times and you'll take the overlap between everything so you can be really confident in a lot of your positions. But there will also always be positions that you can't reconstruct, which we can get into for other reasons.
So Emil, what is the typical timeline of getting a bone in the mail to knowing whether or not you're going to get something really good out of it?
So it depends on the questions you're trying to answer. If you wanted to go super quickly from bone to data, you could probably do it in about 3 weeks, right? If you're like talking, okay, we're going to go in every day, and do really long days in the lab. A lot of that is going to be restricted by the fact that when you're digesting a bone, it takes a week. There's no real way to speed it up. The bone just needs to break down and it takes a long time. Same as when you're doing for radiocarbon, as you're doing those acid washes, you just have to let the bone go until the collagen's there. A lot of the protocols take multiple days. So enrichment will take you about 3 days minimum to do. Library prep will take you about another 2 days. Sequencing usually takes a few days. There are kind of intermediary steps where you can screen and get an idea of how well something will perform. But those are more hit and miss, right? Depending on the questions. If you're trying to reconstruct full mitochondrial genomes, you can get, if you get really good results, you can be pretty confident that it will work, right? But it's a lot of these middling results with samples that aren't, or bones that aren't as well preserved, where you can still recover the mitochondrial genomes, but maybe it will take you 2 to 3 tries. So you get these screening results that are like, and it's only when you take them to the sequencing, you're like, okay, yes, we can do this. We just need to do it 2 or 3 more times, or it's not really worth it. It will take way too much time or money to recover this.
Yeah, I think one of the things the audience might not understand is the amount of time that it actually takes from going from conception of idea, having samples, and then actually publishing results when it comes to this work.
Yeah, so this project, I started it in, I think 2014 or 2015, and I think it was 2014, when we finally published it later this week. So 6 years of collecting, extracting, and doing it. So we have 122 samples in this analysis, 122 individual animals, I should say. But, we're well over 200 to 300 times we had to break these animals down. Some of them, I think like the Mexican Mastodon, we did 9 or 10 times to reconstruct a full mitochondrial genome. Now, I think our stuff scales a little bit better than a lot of morphological analysis on bone. because, for me to process one animal, it takes, 3 weeks, 4 weeks, right? If I were to do it in like a really big time crunch and assuming everything goes correct. But it will only take me maybe an extra hour or two to process another 20 samples alongside of it, right? Versus, so I don't get really this linear increase. If I were to process, I assume like a bone, and I wanted to record all the states for it, doing another bone would cause it to take me twice as long, right?
Mhm.
Versus, because I'm working with very small amounts and very clear liquids, I can multiplex a lot of this through. There are still limits. Obviously, if I'm doing a few hundred, then I have to break it up into multiple sets. But for small increases, I can handle those much better. It's just the problem is with this analysis, a lot of the time you're working with so little data that things never go correct, and then you're ending up having to redo them, or something goes wrong, you don't recover the data you need, you have to go back and try it again, and so on. So this is kind of what we do as a visual representation. If you want to see what this actually looks like on the computer, what I have here are reads. This is from one of the Saltville specimens, and what you can see here is each read is given a name by the computer, and the actual DNA sequence is below it, a spacer, and then we have these quality scores, which are how well the computer or the sequencing machine thinks it called that individual position. I don't remember what exactly these are, the number-wise, because there's a whole matrix that it uses with multiplication and stuff, but we use these later when we're assigning them back to our reference to take positions. We're saying we're more confident in this position than we are in this position, or in this signal for that position than in this one. And you'll typically end up with a few million of these. I mean, the smallest amount of data we take is typically 1 to 2 million reads, and you'll kind of take all of those and try to reassemble them back. On the right is, you can see an example of what that might look like. So you can see kind of our guide at the very top here. In this case, it's an American Mastodon that actually, Chris, you published on. This is the 2016, 2017 underwater archaeology animal. And you can see all of our individual fragments after they've gone through our curation steps lining up. And you can see that in a lot of cases, they're pretty close to what our animal is, but there are, instances like this where we have some damage in the original reads that we recovered that don't match up perfectly. But then you can also see one of the biggest pitfalls with ancient DNA in this stack here. This is actually a very bad example of what you're hoping not to see when you do this analysis. You can notice that here we have relatively even coverage. Maybe there's 10 to 15 reads mapping to a region, and then we have this huge spike, which I've cut off because there's 700 or 800 reads that map to this one region. And because we're working with such small fragments, there's a good chance that sometimes a lot of that residual bacteria DNA that we couldn't remove sneaks through our filters and makes its way into our final alignments. And they map to very conserved regions in the mitochondria. So what we have here is part of the 16S rRNA, which is very similar to some bacterial regions. And we have Mastodon DNA on all sides, and then this huge spike where the bacterial DNA was close enough to our reference that it confused the computer and it placed them into our alignment.
Hey, Emil, I'm going to jump in there. And I'm fascinated to see this, because I remember that specimen. So this was a mastodon tooth that was dredged up by a fisherman off the coast of Rye, New Hampshire. And the fact that you have mastodon DNA in there at all is fantastic.
Yeah.
The fact that you can tease apart potentially. We know that there was probably a fair amount of bacterial DNA in there. When I sampled this specimen, well, I was not very popular in my hallway at the time, because when I drilled into it, it smelled like fish poop.
Oh, well, even better.
Days. It just smelled in the hallway. So we knew there was a lot of contaminants. We knew there was a lot of other stuff in it. So yeah, knowing that there's this section that has just a whole bunch of other stuff, very clearly that fossils don't like to play well.
No, they don't. And we'll see that in the next slide, when we actually get to the outcomes of working on these 122 bones. But it's one of the more challenging aspects that I think people don't realize when we talk about a lot of these bones. Oftentimes there's this fantastic archaeological finds, even going back, 1 million, 2 million years, if you're like, oh, let's get DNA, let's get DNA, let's get DNA. And I'm like, it's already really hard to get DNA out of stuff that's 10,000 years old. As we start pushing that further and further back, it gets rarer and rarer. And even the 10,000-year-old data doesn't want to play well. Going back really far is really hard. But but you know, it isn't impossible. It isn't impossible. So this is kind of the summary of what we got. We got 33 animals in the end out of our original 122, which is about a 27% success rate, which, you know, doesn't seem bad. It's actually pretty good by ancient DNA standards, especially when you consider kind of how degraded and how old some of these specimens are, right? And you can see that generally specimens in the north and Beringia, where they're cold, they're frozen, tend to preserve better, and specimens as we start getting into warmer environments tend to be kind of more variable, right? The DNA breaks down quicker, and so on. But I wanted to really quickly go through some of this Midwestern, Northeastern material, because I think in Chris's words, these are, he says, these samples are basically a hit list for the best mounts in the region. But Chris and Blaine, you're gonna have to jump in here because, like I said, this is your area of expertise more than it is mine. So the first one, some of the first specimens that we had was we got 2 complete animals from Saltville, ETMNH 19334 and 335. I've usually called them as A1A3 and A1A4. This is one of the specimens in the mud pit. I think it's probably under this white tent here, based on some of the coloration I've seen. Maybe Blaine, you might remember.
Yeah, it's actually all the way up here where you see the person in the yellow shirt under the blue
Up
tent.
here?
Yeah, so
Oh, okay.
it's a different time actually when the photograph was taken.
I see.
But that's the area where it's from.
Yeah, but I mean, this is a beautiful tooth. Like I said, I haven't seen too many bones. Most of the time, by the time they get to me, they're small fragments of heavily, you know, either cut or damaged material. But it's a beautiful bone, and I mean, I think it also shows in the data. This is the best or one of the best specimens that we've managed to recover data from. It's one of the highest, what you would say, coverage, good breath, almost complete mitochondrial genome recovery.
We've been really impressed with material from Saltville and just how well that material is preserved when we do try and cut samples for radiocarbon dating or for things like this. It runs everybody out of the lab because it basically smells like burning hair.
Yeah. Yeah, and I mean, that's a fantastic sign, right? If you're getting good collagen material, there's a good chance if you can get good collagen that at least there'll be some DNA preservation. We also have these ones from a bog name that I can't pronounce, but they're a bit south. How do you say it, Chris?
Schimmelpfennig Bog. Yep.
Schimmelpfennig Bog. 2 specimens from here, both in this closely related specimens. I think, Chris, you mentioned these were long bones that you sampled, right? An hour south of, sorry.
Yeah, I sampled long bones from this. These are two mastodons in the University of Wisconsin Zoological Museum. And they are located just south of Madison. And kind of radiocarbon wise, you know, this was part of a big project kind of dating mastodons everywhere just to see when they go extinct. Mm-hmm. These were, for the longest time, we would date things, and then these kept coming out being the youngest mastodons, which was really cool, until about 6 months before we published, and then we had one that was younger. But these are really pretty young mastodons.
Yeah. We also have the Buccaneer Mastodon, of course, this very famous one that's used for the 3D mounted and housed in a new location in the University of Michigan. And these ones from Bonnie Springs, which is, I love this picture here. This is a picture of what I imagine most of your digs must look like. When you guys see that? It just looks awesome and so much fun.
Yeah, that from the 1960s, 1970s.
Yeah.
Chris, I mean, what's the date on that picture, Chris?
71.
Okay.
With David Lindsay. This is a paleontologist that went on to become a professor at a university.
I mean, I imagine he's pretty happy in this one. It looks like there's some great finds. So.
Yeah. This site had mastodons coming out of your ears. 34, 35 mastodons in a single pit, a single hole.
So, Emil, we'll start bringing in questions from the audience in about 4 to 5 minutes.
Okay. All right. Well, so we're running a little bit behind, but I'll go through the next sections a bit quicker. I think we're getting into the results of this paper more now. So, but yeah, this is a specimen that we also recovered that's not actually that closely related to a lot of our Midwestern material, interestingly. So a lot of this is going to be genetic material now. We'll take a very surface level view of it, and if there's questions, I'm happy to answer them. But what you see on the left here is the actual same map that we had before of all of our complete specimens, but now they're not colored just green, they're colored based on their genetic affinity. So you can see that these colors here correspond to these clades, these groupings of genetically similar animals in our tree. And what you notice is this very clear pattern where animals that are geographically close together tend to be more genetically related, which is a fantastic pattern to see. It's exactly what we would expect in proboscideans, given their kind of matriarchal herd structure and at the mitochondrial locus, which is passed down the female line. This is the pattern we want to recover. And that's what we see in elephants and mammoths. But you'll also notice in eastern Beringia, there's this very weird result where we have this large red group, but also this smaller yellow group. And it's actually, they're quite distantly related. So our large red group is down here, and you can see it's more closely related to at least 3 other groups than it is to our yellow group. So what we wanted to do is kind of address this weird blip in our data, this kind of deviance from this pattern that we would expect. And one of the places we turned to was trying to find an ecological explanation for this. So if you remember this kind of summary on the right, both these animals are in an environment where they should really only be there during these warmer interglacial conditions, or at least that's the theory. And because they have a matriarchal herd structure, we kind of can build other genetic assumptions based on that. So if we kind of consider what this model is proposing in context of a single glacial interglacial cycle, it kind of looks a little bit like this, where during these periods of deep glaciation, this northernmost reaches of North America, East Beringia, are these steppe tundra environments. So there's very little food that these mastodons could eat, and they're probably absent from these regions. But as conditions improve, glaciers retreat, and the tree line goes north, these animals can move into these regions from which they were previously excluded and kind of habitate these northernmost regions. But because these glacial interglacial cycles happen on a cycle, and the glacials will come back, these animals are forced with a choice. The ones on the southern boundaries here can probably move south, but some animals will probably choose to go north, and if they choose to temporary refugia, and if that happens, and there's a really deep glaciation, they'll probably die. But you'll also notice that we have our Delta 018 record up here, which tracks glacial to glacial cycles, and you can see that these things are happening, every once in a while, every few 100,000 years. So there's possibility that this is a multiple occurring thing, right? That these animals could be here, then here, then here, and then dying out in most of these patterns. But under such a model, we kind of have 2 big expectations. The first is obviously that animals in eastern Beringia should date to these interglacial periods. And based on this herd structure, if we have a subset of the diversity or subset of the population, the animals in the continental United States moving northward to occupy these regions, we should have relatively low diversity in these northern groups. The first question is deceptively difficult to answer. A lot of the material in Alaska, Yukon is found by miners in the region, so out of stratigraphic context, and it's all past the limits of radiocarbon dating. But what we tried to do in this study is looking at how closely they are related to samples that have radiocarbon ages, kind of estimate their age. And you can kind of see the results of that dating analysis on the right. So 2 different ways of doing a similar thing. And the map that they all were kind of related, the same map, sorry, from before with how they're related on the left. Now this is a little bit difficult, so before we actually get into any sort of patterns, we're going to address how to read this. Each of these is they're known as violins, they're bubbles, and the range in the violin shows you the 95% probability that the age of the animal was within that range. So for instance, you can see this is a sample from Alberta. It has a range from about 350 to 700, right? So there's a 95% chance that the animal's age is within that range. But it also gives us probabilities across that range. So depending on how thick this is, it says there's higher probability in this region than there is in this region of the range. So for instance, this is that same sample under a different analysis, and it's about 150 to 400, but ages around 200,000 are much more likely than ages around 400,000. Make sense? So kind of the big takeaway from this figure is that we have these East Beringian samples in clade Y. So these are these ones in this firm red circle up here. And you can see that they all kind of generally fall around this MIS5 interglacial, which is this shaded gray area here in both analyses. There's a little bit more noise in the joint analysis, but generally they're in this region, and the kind of peak of the probability, the most likely value, always falls within the MIS5 limits. And then we have this orange group up here, which is always older. And these ranges actually don't overlap between these red animals and these orange ones, suggesting that these are pretty concretely 2 distinct time periods that these animals are occupying this region at. And if we kind of put this in context of our tree again, but this time adding a temporal component and overlaying the Delta 018 record at the bottom, you can see that these animals here, as we saw before, kind of fall within this MIS5 interglacial, and these ones in the yellow group fall somewhere earlier in time, probably in a separate interglacial. Although because we're going further back into the tree and there's so much more uncertainty, it's really hard to pin down which interglacial they might fall into. But as we also mentioned, we wanted to look at levels of diversity, and these northern clades, we would expect them to have less diversity than the southern ones that are probably more stable and could live in continuity through these ice ages. So what we have in here is a comparison of all our animals in our large red group and our Midwestern animals, those ones in the large green circle. And essentially, the more blue the intersection between any two animals is, the more similar they are, the less diversity there is between those two animals. And you can see our large red group up here in this triangle at the top is almost entirely blue and very dark shades of purple. There's essentially no diversity within this clade. In comparison, our green clade, our clade G, our central eastern United States clade, has a lot more yellows, lighter purples, showing that there's more diversity within this group than there is in this group. In fact, about 10 times as much diversity within these animals than these animals. So kind of going back to the goals and kind of fitting them now with what we managed to uncover, we first wanted to increase the amount of data, and we do a pretty good job of this, going from 2 mitochondrial genomes published in the last decade for American mastodons to 35 now, which gives us a really nice framework from which new animals that are sequenced can be placed into analyses, or animals from different populations can now be kind of studied in relation to what we know about the rest of diversity on the continent. We also saw that mastodons have this kind of the pattern that we would expect based on their herd structure, but also that these animals in northern environments tend to have lower levels of diversity than the ones in the south. And lastly, we kind of build on these paleoecological models that have been kicking around now for a few years to a few decades, suggesting that mastodons inhabited these northern regions during these warmer interglacials by showing, yes, this is probably the case, but it's also probably not a one-time thing, that mastodons were probably repeatedly moving into these regions every time the climate substantially warmed up. And before we finish, there's kind of, I think, 2 other important things to consider. So the first is, as we know, that these animals don't live in isolation, right? American mastodons are part of their environment, they're keystone species, and they're going to live in those environments with a lot of other species, both plants and animals. And species like camels, beavers that would have occupied potentially similar or the same environment as American mastodons will probably exhibit very similar patterns. Additionally, there's a lot of modern species that are exhibiting very similar patterns. So we see in moose, the northernmost appearance of moose in Alaska is going up by hundreds of kilometers every year. Beavers are rapidly expanding around Alaska into regions in which they previously weren't found. There's numerous birds, fish, and insect species that are also moving northward tremendous distances in very short times, which, in many respects is not a bad thing, in response that they're showing they can adapt to this global warming scenarios and kind of move their ranges. But at the same time, if these animals are experiencing very similar patterns genetically to what we see in mastodons, where these northernmost populations are a subset of the diversity to the southernmost ones, if these southern populations are lost, either because the environment keeps heating such that they eventually get cut off or excluded, or humans expand into that region and decimate their local environments, it could spell kind of long-term genetic consequences for a lot of these animals, making them less adaptable, for any sort of future shifts. And if you haven't seen it, I also wanted to show this image from Julius, which he kind of graciously produced for our paper, showing these mastodons in Beringia and really trying to drive home, these animals would have occupied this environment, but at a very distinct time when the foliage was very different and in very different environmental conditions that we normally see mammoths in. And with that, I'd like to thank, again, the people involved in this paper, both a lot of the collaborating institutions, my colleagues at the McMaster Engineering Center, without which help a lot throughout this entire thing. And of course, the Gray Fossil Site at ETSU for hosting this, inviting me to come talk to you today for slightly longer than I think I was supposed to, but hopefully it was still okay.
Yeah, we threw you a curveball with having extra hosts asking you questions. So
that's fine. I'm happy to talk. If you want me to talk for 5 hours, I can easily talk for 5 hours.
All right. Well, we do have some questions that have come in from our Zoom students that are also here. David, I don't know if you have any other ones that you want to throw in before that or not.
Sure, we've got a handful of questions from our Facebook audience, and then I see I've been collecting the questions from our students here as well, so we'll try to get a few from both sides. I'll start off with a nice, simple, straightforward one for you, Emil. Emil, how do you say your name? I apologize.
Either or is fine. Emil's fine. I usually go by Emil, but it's different depending on the language you use, who's calling, and so on. So.
Gotcha. All right. Well, this question comes from Gino, and it is, did the ancestors of American mastodons originate from Asia?
I mean, a very good question. Chris might actually want to weigh in on this one. Africa, right? I mean, if we're going all the way back, the Mammoth DNA, which is where we have American mastodons, it definitely emerges in Africa, and it's one of the first groups to leave Africa, right? When we're talking about proboscideans. American mastodons themselves, all the evidence points to them being a local evolution in North America. I think some of the earliest well-defined fossils are still about 3 to 4 million years ago, Chris, right?
Yep,
In Idaho and Washington. Although I think there's a lot of taxonomic confusion as to, where those splits are happening, what species do we consider, or what animals do we consider belonging to one species, other species, as we start going back to the record. And of course, as it starts getting more fragmentary, a lot of these questions are more difficult to answer. But if we're talking mastodons in general, not necessarily American mastodons, you're talking about 28 million years ago in Africa.
All right. Blaine, if you want, do you want to take turns? Do you want to pick a student question?
Yeah, if we don't have any other Facebook ones right now.
We've got a whole bunch. I can work through those if you prefer.
I think it's better then to hit the Facebook ones, and we can do the student ones post-recording.
Sure, sounds good. So here's a question. This one is from Donna. Actually, before I ask that question, I just noticed that Carrie has expressed concern that I have disappeared, and that's on purpose. I am the voice in the background at the moment, so don't worry. Donna asks, is the melting of permafrost allowing you to get a big rise in good samples, or are too many samples still not suitable for your purposes?
So, as more bones are uncovered, there's an abundance of samples. Right now, I mean, theoretically, yes, of course, the melting of permafrost will lead us to finding a lot more remains, and I expect if they're found reasonably quickly, before, and I think they will be, otherwise scavengers will probably get to them, they should be pretty good. As it stands, if you want to think about it as a purely numbers game, the field does not have enough researchers to process all of the bones, right? There's one big lab in Canada. Lakehead does also interesting questions, although I don't think they're focusing so much on the Pleistocene. I could be wrong, but I think they're more anthropologically inclined and answering more anthropological questions. And there's a few other smaller labs as we go out further west. But I also don't know what kind of questions they're answering. There's the Shapiro group in UC Santa Cruz, which does a lot of Pleistocene work, but again, mostly focusing on bison, although I think they're branching out now more into other species. I think Pete actually did a lot of his horse work there. So I think it will lead to an abundance of good remains. It's more a question of having the actual time, money, and to test a lot of them, right? And also, I'm not a big fan of just randomly chopping up bones and sampling. The important thing to consider is that this analysis is destructive, right? We were joking before the talk that I don't really consider myself a paleontologist because I don't really see much in the way of bones. Usually when I get them, they're quite small. And the first thing I do is destroy them further, right? That's how my job kind of begins. And that's one of the things we have to be very cognizant about when we do ancient DNA work. Ancient DNA, proteomics, radiocarbons are all and other radiometric means, are very powerful techniques, but they are destructive techniques. So it's very important to, kind of balance the level of knowledge we get from this with also preserving these bones, not just so they're available for other analyses, but also because there's no doubt that these techniques will improve in the future, right? A lot of these bones, even that we did in this study, wouldn't have been possible not even 5 years ago. In fact, if you actually want to see something weird, look into the SOM and just read through some of the supplementary methods. You'll see that there's, even for the extraction of these samples, I think we have something like 10 or 15 different methods, because that's how far our techniques have progressed since we started this project to when we processed that last batch of samples.
What he meant there was the supplemental data that gets published along with the paper. So these types of papers are typically generally short with massive supplemental document that goes along with it.
Yeah, in this case, I think we're well over 100 pages. of additional, tables showing all the reagents, the concentrations and everything. But I mean, even just in the short time I've been in this field, we went from doing phenylchloroform, which is a fairly basic type of extraction, through like 3 or 4 different revisions over the course of this project, right? Each time, hopefully, to liberating a little bit more DNA, improving those methods a bit, and letting us capture older and more degraded material.
All right, here is, let's see, we've got a question. This is from Aaron, so our friend of ours, former student of ETSU. Aaron has more of a general proboscidean question.
Sure.
Why do you think it is that all the lineages seem to lose their lower tusks and develop larger upper tusks?
Yeah, I honestly wouldn't even know where to start.
That's more of a Chris question.
I was going to say Chris might have something.
I can chime in and say we have no idea. It's something that there's a few of us that are working on. And one of the weird things about mastodons is, and proboscideans in general, is that they are just so variable. And that variability can come from so many directions, whether it's taxonomic variability. So it's a variability that is tethered to its biological population, reproductive population, whether that variability is tied to age or sex or just a geographic outlier. And that's one of the real challenges of working with proboscideans. And certainly, even if we just consider mastodon tusks in and of themselves, figuring out why they're there in some cases and why they aren't there in other cases is often not a very straightforward record. The only thing I could say, well, in Gomphotheres, there is some evidence, and Damila Monthe, some of you may have seen her really good talk at the Beringia Center last week. She's done some work on lower tusks in Gomphotheres in South America and finds that you do have some Gomphotheres that lose their tusks, their chin tusks, as they get older. So it seems to be age related. With North American mastodons, a lot of times it doesn't seem to be as clear cut. So Matthew Nabonet is one of our former graduate students who spent a whole semester kind of compiling data on chin tusks of mastodons throughout North America, trying to see if there's any sort of correlation with latitude, any sort of correlation with longitude or age or anything. About the only thing that we could say is that it is you're more likely to retain those chin tusks if you date to an interglacial, so one of these warm periods, than if you date to a glacial period. But it's still not very clear cut.
So that was an excellent question.
Yes.
Because it's one that we don't know the answer to.
Those are the best kind.
Here, we've got a couple more specific detailed questions. This one is from a Mammoth site intern via Charlie. Does the DNA contamination with bacterial DNA and others have any relevant research applications? Do you sometimes get contamination from organisms you want? Can that contaminated DNA be used for anything?
Yeah. Yes, it's so it's important to remember that DNA is a mix-up of environmental contamination and so both from the animal itself and environmental contamination and human. The human one, not very interesting usually. It's been used some cases to make an argument against contamination in human studies. It's not very convincing, in my opinion. As we get more into human material, though, so our lab, I should clarify, is mostly a paleopathology lab. We do a lot more work on ancient human diseases than we do on Pleistocene work. But the Pleistocene material is very interesting because it allows us to do really work and kind of tailor a lot of our questions and a lot of our methods development. And I love it better. The pictures are much nicer when we're talking about presentations, and the stories are much more interesting. When you're dealing with human material, you have that same mix of environmental and bacteria that was within the organism. And from there, you can ask questions about why the animal or why the person died, right? So in addition to recovering the person's own genome, if you wanted to do ancient human studies, you might recover Black Death, which we've done a lot. We have a lot of studies on Black Death recovery. We've done one on smallpox not too long ago, although I think that was from vaccination kits. There's been studies now about Viking smallpox and the like that. A lot of that will be mixed in within that bacterial component. And, we offhand will refer to it as contamination if we don't want it. But you can also screen that for relevant questions. The other application of it is if you have really well preserved material with minimal environmental contamination, and you take samples of the environment alongside the bone, you can sometimes use the differences between them as a strong argument for saying this is probably not contamination and it originates from inside the bone versus this is coming from outside the bone and is more just an environmental microbe.
All right, we've got a couple of questions about other mastodons. So Greg McDonald has chimed in to ask two questions. Have you had chance to sample material from Mexico? And is there any chance of getting DNA from Mammoth pacificus in California?
Yeah, so we do. We have one Mexican mastodon mitochondrial genome in this study. We did 5, I think, total from Mexico. Only one worked, unfortunately, but I mean, that's going to be, a very touch and go region just in general because it's warm. Pacificus is a bit trickier. So if you remember our map, maybe I can actually pull it up here. Let me share back my screen. So if you remember our map of our specimens here, you can see that we did get some regions where this is probably Pacificus. So there's one in California and then these 2 in Idaho. We really kind of would like to fill this out now, especially given, this claims that this is now a separate species. The material is also very difficult though. It's hard to find good preserved region. You'll notice even, in this rare region where we had very good coverage generally, our success rate isn't great, right? Our success rate is inflated in this region, where here we're probably talking about 50, 60, 70% success rates, and it's much lower as we start going further south. So I think it's possible to get Mammoth pacificus material. In fact, we did get very partial material for some specimens that I think are Pacificus. They're just, the data is not there to make any sort of evolutionary, to really answer any evolutionary questions yet.
So David, we're at our 1 hour mark, so we have a decision to make here. We could stop the recording if you want. We could stay live on Facebook to keep answering questions because Emil said he does have time. What do you think?
I'm free till 2, so whatever is easiest for you.
Right. I know that we want to have some time with the students afterwards, and we've got a whole bunch more questions. So I think what we can do is maybe hit a couple more of these questions. And then if there are leftovers, if you or Chris or Emil want to see which comments you might be able to address afterwards.
And we will definitely, if there are Facebook questions that we are not able to answer, we'll definitely get those answered. So, all right, David, if you want to do a couple more.
Let's do a couple more simple ones. One is from Carrie, which I believe is Carrie's daughter, who had to leave to go to class, but we'll answer this question for when they watch the replay. When did mammoths evolve and where did they come from?
Mammoths?
Mammoths, yep.
The mammoth split, I don't know if there's good paleontological evidence on which side of Africa or Eurasia or Asia it happens in, but it's dated to about 5 to 6 million years ago. I think African elephants diverge about 7 million years we have, and mammoths are a little bit, or the split of the ancestor between, the split between Asian elephants and mammoths is somewhere about 5 to 6 million, maybe a little bit older.
All right. And then let's do 2 more. I'm going to do this one. We got one from outside of Facebook. This is from Alton Dooley, whose question is very simple and straightforward. So what's the deal with Alberta?
Yeah. I don't know.
Good question.
Alberta's very weird. Alberta's this very interesting. I think Alberta has a very interesting and a very understudied biogeographic history, right? If we consider, the kind of what Alberta represents, right? Where it's this region of the conflux between these two ice sheets, and it's kind of the first opening and the closing. In some cases where the glaciers don't actually meet, it gives you a corridor between the two regions. It's really weird. We don't have the data where I feel comfortable saying anything particular. What I will say, and and this is why the dating becomes much more important as we start going back. We do have some very weird patterns. We have these three animals that look like they date to three very different periods all from Alberta. So this is an Alberta specimen, and these two blue ones are Alberta specimens, right? They're all found within relatively close geographic proximity, if I remember correctly. They're an hour or two of Edmonton, which is they're all within the circle to the point where I've had to jitter them so they wouldn't all overlap. As to more questions, I don't know. This sample though, again, I don't want to make too many inferences based on a single specimen, especially when the ages of these overlap. But if this does actually turn out that the age is up here and these are in the MIS5, that could suggest that this is an earlier migration event, maybe during MIS7, from a similar or the same source population that eventually gives us these Beringian animals in MIS5. But yeah, Alberta's a very weird region, that we need a lot more data for. I'm hoping to retry some of the Alberta material and see if we can't fill this in. But if I get two more animals and they fall into other parts of our tree, I don't know what I'm supposed to do with them.
All right. Well,
let's do one more. There's one last question that I want to ask because I think this is one that I want to hear the answer to, and I think a lot of people can relate to. This is another one from Aaron, who asks, why haven't we found any frozen mastodons yet? And this is important. The question is followed by a frowning emoji.
Yeah, I mean, it'd be fantastic. So I suspect it's just the environment that they're in. If these animals are up there during warmer, wetter environments, it's probably too long between when they die so when they actually can get solidified in any sort of permafrost. I mean, there's a fantastic blog that I think I saw you link, Chris, maybe on Twitter or on Facebook, about hair specimens and how spotty the mastodon hair evidence is. And I don't feel qualified to. Sorry.
Non-existent.
Yeah. Well, there's the one, right? There's the one hair specimen that was the magnified or whatever. It'd be great just to really kind of solidify that. Mammoths are easy. Mammoths, you go to Siberia, you dig up a mammoth. It's like, oh, we got a mummy. Here's some hair. Boom, end of story. Mastodons is much trickier, and again, it's one of the things I've struggled with. I don't, it's possible that these animals were super hairy in places like Florida and stuff, but I mean, I think it's also pretty possible looking at Columbians, which have much less hair, that they weren't. It'd be nice if they were.
I'm going to jump in here, It's probably more likely that we end up with a bog mastodon, like the bog bodies, where it's just this preserved, dried out husk of an animal.
Yeah.
Although we haven't found.
True. Yeah.
Saltville.
I mean,
there's one there.
Yeah.
Maybe. I mean, in general, I mean, I think the mastodon record is more sparse than it is for mammoths. Wouldn't you say, Chris?
Depends on where you're at.
Okay.
Yeah. So, I mean, there are some places that you just have a lot of mastodons, like the Midwest.
Right.
But yeah, the mammoths definitely do get a lot of attention.
Yeah.
And so that may have something to do with it too. But the other thing is mastodons, we're paying more attention now.
Yeah.
They've gotten more love this week than I think I've ever seen.
Yeah.
Which is good. But focusing on sites like Saltville, where it's kind of a southern site, but you do have this very good preservation in it, and we have. And there may be some of these students who were involved a couple of years ago, and I remember, that there was a clod of dirt that had fibrous stuff sticking out of it, and they said, "What is this? It looks like hair."
Right.
And I said, "Bag it up. We'll take it back. We'll look at it." It turned out to be kind of plant fibers, but we're looking.
Yeah, I suppose the one hope is that eventually the DNA gets so good that it maybe gives us some ideas as to what the phenotypes look like. But that's, I think, pretty far away. There's been some preliminary work done on Wrangel Island phenotypes that has tried to link them. Sorry, to mutations in Wrangel Island animals that has tried to link them to particular phenotypes. But I think that's probably more far away. We're talking at least a few years away, I think. Or someone will really have to devote a lot of time into answering those questions, because I don't know if anyone actually is right now.
Do you think that we can get that kind of coverage of mastodon nuclear genomes?
Yes. I mean, so we run into this kind of problem, right? And a lot of times you can get data. I'm reasonably convinced, if we're going to make a blanket statement, that you can get data from almost any animal, right? The question is always how much time, money, and material are you going to invest into it, right? The Mexican sample to get our mitochondrial genome, we're talking a few grams of material, which is way more than we normally do, or it's probably a few grams at this point. Over 10 libraries, right? So you're talking a fair bit of money, a fair bit of time to get into that animal, right? I'm reasonably confident if you were to even digest a relatively terrible sample and you took the whole animal and melted it down, you'd recover some data, maybe not a nuclear level data, but you'd get some data out of it, right? It's just, you destroy that animal, right?
Right.
Do I think we'll get nuclear? I think nuclear data is very possible without destroying animals. But it's going to be, really being careful and choosing better preserved samples, right? Because already we have in the 2018 paper like a 1x or 2x genome, and I haven't looked at the data for that myself, but I can't imagine it's much better than what we have for some of these other regions in Eastern British and Saltville.
Well, that question was a very simple question that turned out to have a whole conversation worth of answer. So
Yeah.
Thanks to everyone who asked questions, who joined in today. Blaine, I think now's about the time to wrap it up.
Yeah, so again, thank you everybody on Facebook out there. We'll try and answer your questions afterwards. Emil, thank you again, and
Thanks
hope
for
to have
having
you
me.
again sometime soon. All right.
Anytime.
All right.